And finally, Chapter 6, Section 13, protein secretion in the gram-negative systems. Okay. Sec and the TAT, that's going to be more gram-positives. For the gram-negative, since you have that dual membrane, the periplasmic space, you need to get things out across the outer membrane. So, yeah, the sec and the TAT may get things into the periplasm, but it doesn't get it out the cell all the way. So for those, you're going to need secretion systems known as types 1 through type 4 secretion systems. Think of these as glorified protein pipes that span from the cytoplasmic membrane through the periplasm all the way out through the outer membrane. Facilitates, enzyme secretion, DNA transfer, antibiotic release, protein delivery, toxin delivery, exoenzyme delivery, all of it. Type 2, type 1, 2, type 5, type 3, 4, different types of type 6. Notice though, what is it? Type 5 here only spans the outer membrane. The rest of them, 1, 2, 3, 4, and 6, all span cytoplasmic, periplasmic, and outer membrane. 1, 2 are for secretion into the environment. 3, 4, and 6 have been shown for secretion from the bacterial cytoplasm into a targeted cell. Here, cytoplasmic membrane, allowing for targeted delivery of DNA, secreted proteins, which would be an enzyme or toxins. So this is for this, chances are the bacteria is going to be a bit of a pathogen. It's delivering something to damage a cell, which it's then going to cause it to die, burst open, and all its contents now becomes available. 2 and 5 are two-step translocases. Okay, they depend on sec or tat to get stuff to the periplasm, and then the second part is going to move it across the outer membrane. Type 2 is found in a wide variety of bacteria. Okay, this one's widely available. It's widely out there. Transport proteins from periplasm and out. Boom, good to go. Type 5, these are what are referred to as autotransporters. From the cytoplasmic membrane to the periplasm, and then these guys take it periplasm out. Folding drives the secretion. So as the protein's folding, it's being driven or pulled through the outer membrane. So these are going to be proteins that are already in the periplasm. A sec or a tat has taken it from the cytoplasm into the periplasm. It's done some folding, but not completely yet. And when it comes to the final folding, well, as it's going, secondary, tertiary, quaternary folding, it's during that process, it is now traveling out the type 5 secretion across the outer membrane. 1, 3, 4, and 6, well, this is straight cytoplasm out of the cell, crossing all 3 layers of the cell envelope. Type 4 is the one that we see most often with DNA conjugation. This allows one bacteria to interact almost pilus-like and transfer DNA from, you know, donor to acceptor. Type 3 secretion systems, this is ones that we're going to see with toxins and host cells. Allows for damaging delivery of toxins directly into a target cell, a target eukaryotic cell. Here you can see this is what, isolated, these secretion systems actually look like. Purified type 3. So you can see that would be, the parts that would cross the membranes, and then here's where it sticks out beyond the cell.